Dear students, in this video we will deal with the structure of proteins, and the structure of proteins are studied under 4 levels of structural organization, that is primary structure, secondary structure, tertiary structure and quaternary structure of proteins. Each succeeding level is more complicated and is, you can say, a direct consequence of the chemical features of the previous structure. So in this video, firstly we will deal with the primary structure of the protein, which is very simple because it is a primary structure and it mainly refers to the sequence of amino acids in a protein chain held together by peptide bonds. And how we determine the sequence of amino acids? By hydrolyzing the proteins successively by either enzymes or mineral acids, which will tell you the actual sequence of the amino acids in the protein chain. Now proteins are hydrolyzed in various steps, like proteins are first hydrolyzed to proteoses, then to peptones, then to polypeptides, then to simpler peptides and then to alpha amino acids. Amino acid sequence in a protein chain determines its function and is critical to its biological activity. It means that if even you change one amino acid in a protein, it will drastically change the entire physical and chemical properties of that protein. For example, normal hemoglobin, if there is a sequence of amino acids in a protein like hemoglobin, even if you change one alpha amino acid, then the entire hemoglobin will become defective. That is found in sickle cell anemia. Persons suffering from sickle cell anemia possess defective hemoglobin, and the difference between normal hemoglobin and defective hemoglobin is in only one alpha amino acids. Okay, suppose I can tell you here, suppose this is the normal hemoglobin A, B, C, D, just suppose that these are the 4 proteins in that, and in defective hemoglobin you will have the change in only one of them, A, B. Suppose here we include Z and D. So this is a defective hemoglobin and there is a change only in the sequence of only one alpha amino acids. Next is how we will determine the sequence, although it is a very complicated process, but the first method was developed by Frederick Sanger in 1953, in which he has determined the sequence of amino acids in insulin. Insulin has 51 amino acids and it contains of 2 polypeptide chains, one containing 30 amino acids, another 21, and both are linked together by disulfide bonds. Now on the basis of the following generalizations, we can say that primary structure is made up of only alpha amino acids. Secondly, it may contain more than one alpha amino acid chain, usually linked by disulfide linkage, as I have told you already in insulin, discovered by Frederick Sanger. Sequence of alpha amino acids is random and repeating sequences are not common. So repeating sequences you will not encounter much in polypeptide chains or in protein chain. And a small change in the sequence of amino acids drastically change the physical and chemical properties of the protein, that I have already told you in hemoglobin here. And you can see here a fragment or a segment of protein molecule showing the primary structure has been shown. This is the peptide linkage, you can see CONH, and this is one amino acid and this is another amino acid linked together by alpha amino acid linked together by peptide linkage, and this continues and this will give you the primary structure of protein.